बस यही देख रही थी मैं। चेक कर लूँ मैं, उसके बाद शुरू करेंगे। बात ही कर लो। सजीव का फ़ोन तो आया नहीं। अभी 2 मिनट बाद स्टार्ट करेंगे।
मोबाइल पे वैसे भी 2 मिनट बाद ही जाएगा मैडम।
अच्छा, लिंक?
हाँ
तो मतलब लिंक कल वाला लिंक नहीं भेज सकते?
कल वाला ही है मैडम, 2 मिनट बाद चलेगा।
अच्छा, चलेगा ही नहीं?
मतलब अभी नहीं चलेगा, 2 मिनट बाद चलेगा। चलेगा।
कुलजम, वो लिंक मैं भेज रही हूँ, तुम फिर से साजिब को बोल देना ज़रा। उसने कोई रिप्लाई नहीं दिया। आई एम स्टार्टिंग इन 5 मिनट्स, ठीक है? ओके राइट, चलो। मैसेज आया साजिब का, क्या आया? ओके मैम लिखा है, ओके मैम लिख दिया उसने। फिर से भेजूँ मैं? अभी लिंक आया है मेरे पास।
वही कल वाला है मैडम।
कल वाला है? आज उस वही काम करेगा या अब दोबारा भेजूँ?
नहीं नहीं मैडम, वही काम करेगा।
अच्छा,
बोल दो बस कल वाला लिंक है।
हाँ मैंने तो दोबारा कल वाले ही
देखो,
फॉरवर्ड कर दिया।
चेक कर दिया मैंने, कर रहा है काम।
अच्छा, काम कर रहा है,
जी
ठीक
मैम।
है। तो 2 मिनट बाद शुरू करते हैं, स्टूडेंट्स के पास पहुंच जाए, खोलेंगे वो लोग तभी ना।
कल भेजा होगा तो कल वाला ही चलेगा बस।
उनके पास तो फिर होना चाहिए, वही है। म्यूट कर दूं? नहीं, अपनी आवाज़ ही आएगी मुझे।
बस ये हो गया।
हाँ
हाँ, ये कर दो।
इनकी स्टूडियो बंद में।
हाँ, और उसके बाद में एल्टीट्यूड में पहुंचाना।
हाँ
शुरू कर सकते हैं?
हाँ।
अच्छा ठीक है,
देखिए मैम आपका ये आ रहा फोन पे।
ये आ रहा है। हाँ ये अच्छा, ये दिख रहा है और ये लोग दिख रहे हैं।
हाँ तो आप स्टार्ट कीजिए ना।
आप start करेंगे।
हाँ, start कर दीजिए।
स्टूडेंट में आगे आपके पास दिखता है लाइव चैट में?
नहीं
जी,
मेरे
जी।
उसमें लाइव चैट का ऑप्शन नहीं आता मेरी
मेरे
साइड
में तो आ रहा है।
हाँ,
जी,
ठीक
आप-
है,
इसका
ठीक
मतलब ये स्टूडेंट्स आ गए
जी
हैं।
आ गए
मैम ये
हैं।
स्टूडेंट्स आ रहे हैं।
7 स्टूडेंट्स हैं आपके पास ऑनलाइन में।
ठीक है, और 2 बजे से 2:03 लिखना शुरू
कर दीजिए।
करो। ठीक है, दरवाजा
ये देखो मैम आपकी आवाज़ भी आ रही है इसमें।
ठीक है, ठीक है। So very good afternoon to everybody. Today we are going to have class on B cell development and activation. So I would like to tell one thing, that yesterday we had class on antigen processing and antigen presentation, and I think 1 or 2 student has some problem in cross presentation, and that time the lecture was over. So I would request you to come to my room anytime and we can discuss in detail, because by the time the live chat was over. So today we are going to discuss about B cell development and activation. So after the class, you should be able to know the development of B cells, B cell activation and proliferation with thymus dependent and thymus independent antigens. And you should always be, also be able to explain the various antibody mediated effector functions and difference between primary and secondary immune responses and B cell related clinical disorders. So when we talk about B cell development and the activation, we know that we have 2 kinds of immune responses, humoral immune system and the cell mediated immune system. Interaction of B cells with antigen and their subsequent proliferation and differentiation into antibody secreting plasma cells, that is the humoral immune system, whereas cell mediated immune system involves T cells. So when we discuss about B cell development, what do we know about B cell? B cell are developed in bone marrow, so hence the name is given B cells. The production of plasma cells and memory cells can be divided into 3 broad stages. The number first is maturation, number 2 is activation, and third is differentiation. The maturation or the generation of mature immunocompetent B cells, whereas in activation, the activation of mature B cells when they interact with antigen. The differentiation of activated B cells into plasma cells and memory cells. So first the cells will mature and they become immunocompetent B cells. Then they will be activated when they interact with the antigen, and finally the differentiation of activated B cells will take place into plasma cells and the memory B cells. So what happens in maturation? The generation of mature B cell first occurs in the embryo and continues throughout life. Before birth, the yolk sac, fetal liver and fetal bone marrow are major sites of B cell maturation. But after birth, the generation of mature B cells occur in bone marrow. We have progenitor B cells which proliferate in bone marrow, and these progenitor B cells are also known as pro B cells, and they express a transmembrane tyrosine phosphatase. You can call it CD45R or VLA4, that is very late antigen 4. So these progenitor B cells which proliferate in the bone marrow expresses a transmembrane tyrosine phosphatase. The proliferation and differentiation of pro B cells into precursor B cell or pre-B cell requires the microenvironment, and this microenvironment is provided by bone marrow stromal cells. So how the stromal cells can provide some microenvironment? These stromal cells play 2 important roles. Number 1, they interact directly with progenitor B cell and precursor B cells, and secondly, they secrete various cytokines, and specifically IL-7 support the developmental process. So these stromal cells which are present in the bone marrow, they provide the microenvironment where they directly interact with progenitor B cell and precursor B cell and also secrete IL-7. So in this picture you can see that this is the bone marrow stromal cell and we have progenitor B cell. The progenitor B cell has VLA4 as the surface receptor, whereas on the stromal cell we have VChem1. So there is an interaction between VLA4 and VChem1. Once this interaction takes place of progenitor B cell and stromal cell, then one more c-Kit molecule start expressing on progenitor B cells, and on the stromal cell we have SCF or stromal cell factor. Then they have the interaction. So once we have these two interaction, VLA4 and VChem1, and secondly c-Kit and SCF, then these stromal cells, they start secreting IL-7. Also, when there are interaction of these two, then these progenitor B cells, they starts expressing IL-7 receptors. So once receptors are there on the progenitor B cells and stromal cells, they are secreting IL-7. This IL-7 goes and bind to these receptors. Once these IL-7 bind to this receptor, now this progenitor B cell further mature into precursor B cell. So now this precursor B cell further proliferate into this mature B cell where we have membrane bound IgD or membrane bound IgM. There is core expression of co-receptors, membrane IgM and IgD. So now you can see here that how the development of B cell in bone marrow takes place, and the stromal cells play important role. They produce certain receptors or the ligand for the receptor of present on the progenitor B cell, and the interaction leads to the expression of IL cell receptors on these cells and IL-7 secretion by the stromal cells. So during the development of B cells or maturation of B cell, there are different steps. We have progenitor B cells, then large progenitor B cells where the gene rearrangement starts of the heavy chain first. Heavy chain, we know, you must have learnt about the gene rearrangement in the immunoglobulin genes, where in the variable region we have VDJ. So in the variable region there is a rule that D will first join with the J and form a DJ segment, then only any of the variable gene segment will come and bind to DJ. So the second step is large progenitor B cell, the size increases. Then progenitor B cell have expression of VDJ or the heavy chain variable region is expressed. Then there will be starts the expression of Ig alpha and Ig beta. You must be knowing that these B cell surface receptors like immunoglobulins, they don't have signal transduction sites, or they don't have intercellular like chains so that they can pass on the signal. Therefore, the Ig alpha and Ig beta plays important role for the signal transduction in the B cells. So the Ig alpha and Ig beta starts expressing, and these precursor B cell now starts this expression of the light chain also, that we call surrogate light chains initially. Then finally in the immature B cell we have the expression of IgM, and then we have Ig alpha and Ig beta. Why IgM first? Because when we see the DNA sequence in the immunoglobulin genes, in the constant region mu is the first one and delta is the second, and there is no switch side between M and D, hence whenever there is an expression of IgM, there will be co-expression of Ig2 when an immature B cell is now going for the mature B cell. So mature B cell which have IgM as well as IgD expression or the co-expression of both these two make the immature B cell to mature B cell, which is, we call it, immunocompetent naive B cell. Immunocompetent naive B cell means it has not come across any antigen, but it can be further activated on receiving any of antigen. So this mature B cell which have co-expression of IgM and IgD is an immunocompetent B cell. So just to recapitulate that immunoglobulin gene rearrangement produces immature B cells first. The B cell maturation depends on the rearrangement of the immunoglobulin DNA in the lymphoid stem cells. First to occur is the pro-B cell stage, is a heavy chain gene, DJ gene rearrangement, then V to DJ gene rearrangement, and cell is classified as a precursor B cell. Then a precursor B cell goes into an immature B cell, requires a productive light chain gene rearrangement. An immature B cell express membrane bound immunoglobulin M only, IgM, on the cell surface. Then B cell development culminates in the production of an IgM bearing immature B cell. At this stage the B cell is not fully functional, so full maturation is signaled by co-expression of IgM and IgD on the membrane by differential RNA processing. So this should be clear that an immunocompetent cell have co-expression of IgM and IgD. So here we can see that overall B cell development, what is happening, they have two phases, antigen independent phase and antigen dependent phase. In the antigen independent phase, the progenitor B cell, which has a surface marker, that is the tyrosine phosphatase, VLA4 or CD45R, this progenitor B cell upon gene rearrangement convert into mature B cell, and which is having the immunoglobulin receptors on the surface. At this stage also there is a selection process where negative selection takes place, autoreactive antibodies, self-reactive antibodies, the cells which express that will die off, and these mature B cell will come out of the bone marrow and reaches to the peripheral lymphoid organ. In the peripheral lymphoid organ, you will see here that these cells are naive B cell, but they are immunocompetent B cells. So if we are having approximately 5 into 10 to the power 6 cells per day which are reaching to the lymphoid peripheral lymphoid organ, these naive B cell can either come across of any of the antigen, or if they are not come across of the antigen, they have a short lifespan, and over a passage of time they will be apoptosed. So you will be surprised to know that out of 100% cells, 90% cells gets apoptosed and die off. Only approximately 10% cells which are exposed to any antigens and got some T helper cell help, they gets activated, and then further on class switching, on affinity maturation, they can form plasma cells and memory B cells, and these plasma cells can secrete the antibodies. So now the overview says that yes, in antigen independent phase the progenitor B cells go into the mature B cell which have co-expression of these IgM and IgD, and they are immunocompetent B cells. When they go into the peripheral lymphoid organ, then they are naive B cells, and when they come across of the antigen, they can be activated and then they can produce antibodies. So as I was telling, the self-reactive B cells are selected against in the bone marrow. It is estimated that the bone marrow produces about 5 into 10 to the power 7 B cells per day, but only 10% are actually recruited into the recirculating B cell pool. 90% of the B cells produced each day die without ever leaving the bone marrow, and negative selection and subsequent elimination by clonal deletion of immature B cells that express autoantibodies against self-antigen in the bone marrow are dying off. So how these B cells gets activated and proliferate? After export of B cells from the bone marrow, activation, proliferation and differentiation occur in the periphery where they require antigen. These antigen-driven activation and clonal selection of naive B cells leads to the generation of plasma cells and memory cells, as we have seen in the last picture. But in the absence of antigen-induced activation, naive B cells in the periphery have a short lifespan, dying within a few weeks by apoptosis. So this picture again recapitulates that in the bone marrow the stem cells are there. Then after gene rearrangement, these cells are maturing to mature B cells, the immunocompetent B cells. And once these B cells come across of any of the antigen, they proliferate and then they differentiate into memory cells and the plasma cells. These plasma cells produce further antibodies. So far we have learned about maturation, and now we are going for the activation. How these B cells gets activated, what kind of antigens can activate these B cells. So we have two kinds of antigen, thymus dependent and thymus independent antigen. So depending on the nature of the antigen, B cell activation proceeds in two different routes. The B cell response to thymus dependent antigens requires direct contact with T helper cells. As the name suggests, thymus dependent antigens means direct contact with T helper cell, not simply exposure to T helper cell derived cytokines. If we provide cytokines but no T helper cell physical contact or direct contact, then thymus dependent antigen will not activate B cell response. But if an antigen that can activate B cell in the absence of this kind of direct participation by T helper cells are known as thymus independent antigen. So one is thymus dependent where they require direct contact with T helper cell. The other one is thymus independent where this kind of direct participation is not required. So again this flowchart tells that B cell activation can be by thymus independent antigens or by thymus dependent antigens. The thymus independent antigens can again be of two types. Type 1 thymus independent antigens TI1 or type 2 thymus independent antigen TI2. This is TI1 and TI2. Thymus independent, the classical example of thymus 1, type 1 thymus independent antigens or TI1 is lipopolysaccharides. Whereas type 2 thymus independent antigen can be bacterial flagellin or the bacterial cell wall or other polysaccharides. Whereas thymus dependent antigens require direct contact with the T helper cells. So first to discuss about TI1 antigen, thymus independent type 1 antigens. So most TI1 antigens are polyclonal B cell activators or mitogens. At high concentration, let us say LPS, at high concentration, some TI1 antigen will stimulate proliferation and antibody secretion by as many as 1/3 of all B cells. Whereas at low concentrations, only those B cells specific for epitopes of the antigen will be activated. So at high concentration, it acts as polyclonal B cell activator, and at low concentration, it can have B cell specific immune response or B cell specific response can be there. The stimulate antibody production in nude mice also, it means it does not require T helper cell help at all. That's why we call it truly T cell independent, neither the cytokines nor the physical contact of T helper cells. When we talk about the second type, the TI2 antigen or type 2 thymus independent antigens, they activate B cells by extensively cross-linking the membrane immunoglobulin receptor. Let us have, we have a bacteria where we have many epitopes. So they will cross-link with the membrane immunoglobulin receptor and in nude mice which lack thymus derived T cells, we are not having thymus derived T cells there, but do contain a few T cells in intestine. These Ti2 antigens do elicit B cell response. What does it mean? In type 1 they were truly independent, even not required the cytokines, but in type Ti2 type they required certain B cells, T cells. If they are present in intestine, they can produce some cytokines which can help to elicit the B cell responses. Administration of few T cells to TCR knockout mice restores their ability to elicit B cell responses to Ti2 antigen. So if you knock out a mouse where we don't have any T cells anywhere, then there will be no response. But if we provide few T cells, then B cell response to Ti2 antigen can be elicited. So this picture very clearly tells you that number 1, on the upper side, the A, the B cell responds to high concentration of LPS, Ti1 type antigen. So when the concentration is high, what is happening? Then these plasma cell can produce polyclonal IgM response, different kind of IgM response, the polyclonal response. Whereas if we have low concentration of LPS, then it is limited, the LPS is limited, what will happen? It will only elicit antigen specific activation of that particular IgM. So antigen specific IgM will be generated. So it will be the specific B cell response when the concentration is low, but when the concentration is high of LPS, these Ti1 antigen can generate polyclonal response. The other type, the second type, Ti2 antigens, where these B cell can produce IgM specific, and in certain cases it can occasionally have class switching. So from IgM it can have certain amount of IgG, and which occasional to nil memory cells are not developed in this, and it requires certain help from the T helper cytokines. That's why if even in intestine some T cells are present, they can produce some cytokines which can help to generate this response. So I hope now the Ti1 and Ti2 responses are clear to you. So again, now we can compare all three, the thymus dependent antigen, thymus independent antigens type 1 and type 2. So in thymus dependent antigen, the chemical nature is soluble protein, whereas in type 1 it can be bacterial cell wall components like LPS, or it can be polymeric protein antigens or capsular polysaccharides in type 2. When we talk about humoral response, it can be isotype switching, affinity maturation, immunological memory, and polyclonal activation. These are the important features of humoral response. So in thymus dependent antigen, isotype switching is there. It can generate different immunoglobulin response, IgM, IgD, or IgE, or IgG. Or in the type 1 response, the isotype switching is not there, and type 2 very limited isotype switching is there. When we talk about affinity maturation, yes, it is present in thymus dependent antigens, but not in type 1 and type 2 thymus independent antigens. Immunologic memory, yes, it is there with thymus dependent antigen. It is not there with type 1 and type 2 thymus independent antigens. When we talk about polyclonal activation, it is not there because in thymus dependent antigen there is a specific response. So no polyclonal activation, but yes, in type 1 at high doses it gives the polyclonal this activation, where in type 2 it is again specific, not the polyclonal activation. So these are the basic differences. You should remember that yes, how these thymus dependent and thymus independent antigens can activate the B cell. So once we have these antigens which can derive these B cells by different signals, The T dependent antigen, now thymus independent we have learned, now thymus dependent antigens, how they derive B cells to the activation of this. So the competence signal is there, two types of signal, the competence signals and the progression signals. So T dependent antigen derive B cells by competence signals and the progression signal. The competence signal drive the B cell from G0 into early G1 phase, whereas progression signal from G1 into S phase and ultimately to cell division and differentiation. So both the signals are important because first drive B cell from G0 into G1 phase and the progression signal derive G1 into S phase and finally to cell division and differentiation. The competence signal is achieved by two distinct signaling events, signal 1 and signal 2. What are these two signals? Once the B cell has acquired an effective competence signal, signal 1 and 2, in early activation, the interaction of cytokines and other ligands with the B cell receptor provides progression signal. So first signal is coming when there is an effective competence signal in early activation, and then interaction of cytokines and other ligand can give you the progression signal. So two types of activation signals are there. Binding of a thymus independent antigen to a B cell provides both the signal. So thymus independent antigen just by binding of the antigen to the B cell provides both the signal, signal 1 and signal 2. Whereas a thymus dependent antigen provides signal 1 by cross-linking membrane immunoglobulin and separate interaction between CD40 on the B cell and 40 ligand on the activated T helper cells is required to generate signal 2. So what does it mean? That yes, thymus independent antigen provides both the signal just by binding of antigen to the B cells. Whereas in thymus dependent antigen, signal 1 is provided by cross-linking and second signal is coming from the stimulatory signals or co-stimulation, CD40 on the B cell and CD40 ligand on the activated T helper cells. So here in this picture you can see that thymus independent antigen, both the signals are coming just by cross-linking the antigen to these membrane immunoglobulin receptors. The antigen is bound to the immunoglobulins and then the signal is sent by, both the signals are sent by the co-receptor, that is Ig alpha and Ig beta. Whereas in thymus dependent antigens, the binding of antigen to these immunoglobulin receptors sends signal 1 and signal 2 is coming by these co-stimulatory molecules CD40 on the B cell and CD40 ligand on the T helper cells. Then they have different steps. What are the different events going on just by activation of the B cell by attaching the antigen to the B cell and then till the proliferating B cells which are providing plasma cells and finally the immunoglobulins. What is happening? So in first event, what is we discussed that yes, signal 1 is coming just by binding of antigen to the immunoglobulin receptor. So antigen cross-links the immunoglobulin receptor generating signal 1, which leads, the signal 1 leads to the increased expression of MSC class 2 and co-stimulatory B7 molecule, also CD40 molecule on the B cell. So just by binding of antigen to the receptor, signal 1 is there, which can give the signal to express more and more MSC on the surface, then 40 ligand and B7. The antigen antibody complex are internalized by receptor mediated endocytosis and then degraded to the peptides. We know yesterday we have learned that yes, the receptor which is binding to the antigen is just being taken up by endocytosis, that is the membrane clathrin coated pits, and then they go to the endosomal pathway, first to the early endosome, then to the late endosome and finally to the lysosome. And what is happening? Now this antigen is processed, degraded in small pieces of desired length and then it is being loaded on the MSC2, and which is coming on the surface and presenting this antigen to the T cell. So some of which are bound by class 2 MSC. After processing, the antigen is processed and presented on the MSC surface. That is a peptide group of MSC. It is presented to the T cells. So in the second step, what is happening now? The T helper cell recognizes antigen class 2 MSC on B cell surface, and this T cell receptor recognizes this particular antigen. By recognizing this, what is happening? The B7 and this T helper cell start expressing CD28, and there is a co-expression of B7 and CD28. This plus co-stimulatory signals activates the T helper cells. Once the T helper cells gets activated, they starts expressing CD40 ligand, and CD40 is already there on the B cells by signal 1 only. So what is happening? The T helper cells begins to express CD40 ligand. Interaction of CD40 and 40 ligand provides signal 2. Signal 1 is coming after binding of antigen to the receptor, and then having different events where expression of MSC, expression of CD40, expression of B7 is there. Once the antigen after processing is presented to T cells through TCR here, then there is a co-expression of CD40 as well as CD28 on the activated T helper cell, and this interaction of CD40 40 ligand sends signal 2. So once the B cell begins to express receptors for various cytokines, after having this signal 2, they starts expressing the surface receptors of cytokines, and on the other side the activated B cell, they start secreting the cytokine, let us say IL-2, which goes on binding to the IL-2 receptor on the B cell. So after binding of cytokines released from the T helper cells in a directed fashion, sends signals that support the progression of B cell to DNA synthesis and to the differentiation. So as we learned, the G0 to G1 phase, and G1 to S, and then to cell division and further proliferation of B cells, and then there will be formation of plasma cells and antibody secretion. So the events are not so simple as we are discussing. We know, as I have already discussed, that B cell receptors, whether it is IgM, IgD or IgG, they don't have signal transducing molecules inside the cytoplasm. They don't have any. So what is happening? They are binding to the antigen, but how these signals are sent? These signals are sent only by IgG alpha and Ig beta. So signal transduction is being done by Ig alpha and Ig beta, which have item motifs. What have item motifs? Item motifs are nothing but they are immunoreceptor tyrosine based activation motif or items. They have tyrosine molecule, and upon activation they start phosphorylating these tyrosine molecule. Different enzymes are present, and they start kinases, they start phosphorylating these tyrosine molecule, and hence further activate the downstream molecule of the signal transduction pathways. So this way the co-stimulatory molecules are the co-receptors are very important on the B cell membrane. Not only Ig alpha and Ig beta which have items, there are certain other co-receptors are present like CD19, which also has phosphorylation sites and the tyrosine molecule, as also the CD21, which recognizes C3D molecule, which is a part of complement pathways. C3D molecule is being recognized by CD21 receptor, and these all together, the Ig alpha, Ig beta, CD19 and CD21, they are all co-receptors and helping these role of B cell co-receptor complex in enhancing the activation signals. And again you can see that with this picture also, Okay, these are not so simple. These B cell activation and this signal transduction can induce many pathways, many molecules like calcium, calmodulin, and then PKC, phosphokinase C, which can activate NF kappa B, or RES or MAP kinase cascade, or even the PI3 kinase, which can give the signal of cell survival, and then further the activation of the gene here, so that more and more proliferation will be there. So far what we have learned the B cell, how the B cell is matured, how the B cell is activated, and how they are differentiated to plasma cells and the memory cells. These plasma cells, they start producing the immunoglobulins. Once they have produced the immunoglobulins, by just recognizing the antigen by this antibody is not sufficient to have the immune response. Immune response will be there, but effector functions are certainly required, which are antibody mediated. Effector functions means once the antigen and antibody interaction is there, then finally this antigen has to be cleared off, or the killing of this particular pathogen which an immunoglobulin has identified. So antibodies participate in different biological activities, and killing or elimination of pathogen is not solely by binding to antibodies. Antibodies invoke responses, and these effector functions result in removal of antigen and killing of that particular pathogen. Which part of immunoglobulin is conferring this effector function? We know we have variable regions and we have constant region in the heavy chains. The variable region of light and heavy chains is forming the antigen binding site on the immunoglobulin, whereas heavy chains constant regions responsible for variety of collaborative interactions with other cells, and hence performing the effector functions of the humoral responses. So the heavy chains constant region, we have CH1, CH2, CH3 like different domains, constant domains in the heavy chains, which result in the effector function, and we have the humoral response. And we should also know that not all classes of immunoglobulin have same functional property. Some have better complement fixation, better complement activation, others are good opsonins, and some are performing ADCC effect. So we have some important effector functions. So the important one is complement activation. You have a separate class on a complement, so I will not go into detail, but just to brief you that many immunoglobulins and their subclasses activate the complement pathway. You must be knowing it that IgM and IgG3 subtype of IgG are the best complement fixers. They are the good complement activator, and they are the main antibodies which activate the complement through the receptors present on their Fc region. This system generates inflammatory mediators and promotes microbial phagocytosis and lysis. They also initiate by the binding of a complement protein called C1q fragment to the Fc portion of the antigen complex with the immunoglobulin G or IgM, because when the antigen antibody complex is there and then these complement fragments they are attaching, finally they form a MAC, membrane attack complex, which makes pores into these plasma membrane of these pathogens. When the pores are formed, all these solvents or the cell's material is going inside the cell, and hence it can be osmolized and it will die off. So this way the complement activation is the important effector function of humoral response. Here I have put one picture where these let us say any bacteria is there which have many antigens on the surface. The immunoglobulins has recognized these antigens, and then this is the Fc region. This Fc region will bind to the phagocytic cells which have Fc receptors. This will bind to the Fc receptor, and again the CR1 receptor, complement receptor 1 is present, and here we have C3, and these C3b will also bind to CRB. So this way this complex will be formed, which will be phagocytosed, and then the pathogen will be killed by different methods like oxidative damage and some enzymatic degradation. The second important effector function is opsonization. The promotion of phagocytosis of antigens by macrophages and neutrophils. Surface receptors of macrophages and neutrophils have Fc receptors which can bind to the constant region of the immunoglobulin main in the heavy chain. Okay. Binding of FCR with antibody complex with the target or the bacterial cell helps in binding of the pathogen to the phagocytic membrane. Results in the phagocytic pathogen killing targeted by antigen enzymatic digestion and oxidative damage. Thus, antibody function as opsonin and enhances phagocytosis manifold for particulate antigens. So, these particulate antigen where the opsonization is taking place as the effector function, the mechanism is more or less same as we have just learned in the complement activation. What is happening? We have any bacterial cell, let us say it has many antigen which has been covered by the immunoglobulin, recognized by the immunoglobulin surface receptor, and then these cells, the phagocytic cells, let us say macrophage, they have the Fc receptor. So the Fc portion is recognized by the Fc receptor, and the whole complex is taken up by the these macrophages, digestion in the lysosome, and then there is an enzymatic degradation having the low pH and oxidative damage, and where the cells can, this pathogen can be killed. So coating of antibodies and bacterial cell renders it recognizable as foreign by phagocyte such as macrophage and neutrophils, which then ingest it and destroy it. Then we have antibody dependent cell mediated cytotoxicity. Here O is missing, sorry. So the ADCC, antibody dependent cell mediated cytotoxicity. So what happens in ADCC? There are number of cells which have cytotoxic potential expressed on the membrane receptors for the Fc region of antibody molecule and kills the target cell. So many cells have cytotoxic potential which express on the membrane receptors for the Fc region of antibody molecule and kills the target cell. When antibody is specifically bound to the target cell, receptor bearing cells bind to Fc region of antibody, causes lysis of the target cell by releasing cytotoxic substances like TNF alpha, like perforins, granzymes, etc. NK cells, macrophages, monocytes, neutrophils and eosinophils, they can mediate the antibody dependent cell mediated cytotoxicity. Although non-specific for antigen, but antibody directs them to the specific target, and this cytotoxicity referred to as ADCC. So it will be more clear with the picture where we have a target cell and it is being the antigen is being recognized by the immunoglobulin, and the Fc region is bound to the Fc receptor on the cytotoxic T cell, cytotoxic cells like NK cells, neutrophils, eosinophils. These cells, they have cytotoxic material. So once the target cell is bound to immunoglobulin and Fc region is recognized by Fc receptor, these cells gets activated and start releasing their cytotoxic material like perforins, like TNF, some enzymes and other molecules. And these molecules then they non-specifically target this cell, and there is a cell cytotoxicity and cell die off. This we call ADCC. So we have covered the B cell development, then we have covered the effector functions which are mediated by the immunoglobulins and in the cause of the humoral response. So this humoral response which is generated can be primary and secondary. We have two kinds, primary and secondary. All of you know that why we have vaccination so many times, why we have booster shots. So the primary humoral immune response is the antibody response to an initial antigenic stimulus or first contact with the exogenous antigen, which generates primary response. So the antibody response which is generated by first contact of that particular antigen. The secondary response is the response to the subsequent stimuli with the same antigen. When you get exposed to the same antigen again, then secondary response is generated. So what is the difference between primary response and the secondary response? In the primary response, we have a lag phase. The naive B cells undergo clonal selection, expansion and differentiation into plasma cells and memory cells. So it takes time for the naive B cell when they first contact with their antigen for the first time, then the plasma cells and memory cells are formed. So there is a lag phase of 4 to 6 days initially to have all this, and the peak response reaches between 7 to 14 days. But what happens in the secondary response when the same antigen, let us say antigen A, which was given first time, if a person is exposed to the same antigen A again, then there is a very small lag phase or no lag phase, and the response reaches to the peak in a very shorter time and depends on the persistence of the antigen, and it has much heightened response. Here the serum antibody level is just here. Here you can see in the same with the same antigen, the secondary response is much more heightened or exaggerated. So when we want to differentiate these two response, important point, always ask in the Viva, what is the, just mention the difference between primary response and secondary response. Then we have to mention few important points. Yes, responding B cell in primary response is naive B cell. It has not come, although these cells are immunocompetent B cells, but they have not come across to that antigen earlier. So these naive B cells which get first contact to that antigen, and then, but in the secondary response we have memory B cells present. Okay, because in the primary response we have plasma cells as well as memory B cells are generated. The lag period following antigen administration is generally 4 to 7 days in the primary response, whereas generally 1 to 3 days in the secondary response. Time of peak response is 7 to 10 days, whereas in secondary response just 3 to 5 days. So shortened time of response. Then magnitude of peak antibody response, it varies depending on the antigen, what kind of antigen is there, then the magnitude of antibody response will be there. But the same antigen in the secondary response generally can induce 100 to 1000 times higher response than the primary response. That's why it is a heightened response, or we call it exaggerated response. The isotype produced in primary response is IgM, which predominates early in the response, and later on in the secondary response we have IgG, which predominates. The antigens, if they are thymus dependent and thymus independent, they can have primary response, but secondary response generally thymus dependent antigens have much good secondary response. The antibody affinity, it is lower in primary response and it is higher in the secondary response. So these are the important differences between primary and secondary response: the responding B cell, the lag period, the time of peak response, the magnitude of antibody response, the isotypes produced, and the antibody affinity. So one more thing, it always tells me that how these memory cells, how do they decide that these cells will go for memory B cell formation and these cells will go for the plasma cell formation. So I have gone through some reviews, and then we found that yes, in one of the reviews they has mentioned that when the B cell gets in contact with the antigen and some T cell help is there, they always gets activated, and then they form the germinal centers, and these germinal centers they have dark zone and the light zone. And there are certain molecules, certain proteins, certain factors are present which are actually important. It is not just like antigen is the B cell gets exposed to an antigen and then the B cell gets activated and there is a formation of memory cells in plasma. There are many events in between, many factors which are involved, because when they receive help from cognate T cells, if the B cell express the appropriate molecules such as combination of BCL6, which is B cell lymphoma 6, the ICOS ligand, that is inducible T cell co-stimulatory ligand, or the BLIMP1. BLIMP1 is the B lymphocyte induced maturation protein 1. So the expression of these molecules actually decides, yes, that these B cells which gets activated will produce, yes, plasma cells and memory cells. So when they go into the germinal center and they cycle between centroblast and the centrocyte, these B cells, if they have activated B cell, if they have higher expression of BLIMP1, they will certainly form plasma cells. If BLIMP1 is low, those cells can go for memory B cell. And these are the markers for memory B cell, which can definitely tell you, yes, these cells are memory B cells. Although the expression of BLIMP1 is crucial for the formation of plasma cells, the factors that control memory B cell formation are less well defined, still not fully defined. So these memory B cells which are formed again compete for the higher affinity, because memory B cell pool is fixed. So in second picture of the same review, we can find out that in the germinal center, these cells which migrate from the germinal center throughout the immune response as either plasma cells or memory cells, under the influence of B cell receptor stimulation, these centrocytes with high affinity for antigen differentiate preferentially. So high affinity cells will differentiate preferentially, and they can form the plasma cells or they can form the memory cells. So if one memory cell is there, not necessarily all the cell will survive, they can be apoptosis. So only prolonged survival, and we should know the size of the memory B cell pool is finite. There's no, it's not like that plasma cell number is 10 and memory cells will be 8. No, the size of memory B cell pool is finite, so the survival of memory B cell is competitive. Prolonged survival in the germinal center correlates with increased affinity for the antigen, improves the competitiveness, and this competitiveness also known as fitness of the memory cells. So high affinity, high fitness antibodies, high fitness cells will survive as memory cells, and high affinity cells will survive as the plasma cell. So this is important, how these memory cells and this fitness of the memory cells is naturally the which form the finally the memory B cell pool, and the increasing intensity is this picture shows that they are of the high affinity and high fitness. So far again, so primary secondary response has been covered. You have seen in many journal clubs that B2 cells and B1 B cells and marginal zone B cells. So the difference should also be clear to all of you, that follicular B2 B cells are the conventional B cells which express CD19 or CD21. They have co-expression of IgM and IgT and also the expression of CD23. Whereas B1 B cells have IgM expression and CD5 expression. Whereas marginal zone B cells have 19 and 21 IgM, but instead of CD23 they have CD1 expression. So how they are different? What are the major sites where they reside? Follicular B2 cells, B1 cell, or marginal zone B cells. Actually, conventional B2 cells were so named because they developed after B1 B cells. That's why their name was given B2 B cell. Although these are the conventional B cells which are generally the major site is secondary lymphoid organs. So when we see the difference between these three, follicular B2 B cell, conventional B cell, B1 B cells, and marginal zone B cells. The major sites for these cells, for first the follicular B2 cells, is secondary lymphoid organs, whereas B1 B cells, they reside, or the major site is peritoneal and pleural cavities. The marginal zone B cells reside in the marginal zones of spleen. The source of new B cells from precursors in the bone marrow, whereas these B1 B cells are self-renewing division of existing B1 cells, and they are long-lived, may be self-renewing, not yet sure. V region diversity, that the variable region diversity, very highly diverse, as you have seen antibody diversity 10 to the power 6. So these cells, conventional B cells, are highly diverse, whereas restricted diversity in B1 B cell and somewhat restricted in marginal zone B cells. Somatic hypermutation. Somatic hypermutation is one of the feature of these antibody diversity. Yes, they are present in conventional B cell, but no hypermutation in B1 cells, and it is not still clear whether marginal zone B cells have hypermutation or not. Requirement for T cell help. Yes, B cell require T cell help, but B1 B cell does not require, and it is variable. Sometimes they may require, not require. Isotypes produce high levels of IgGs produce, and other subtypes also, but here in B1 B cell the high levels of IgM is the one, and primarily IgM and some IgG in the marginal zone B cell. The response to carbohydrate antigens. Generally the conventional B cells respond to the protein antigens or peptide antigens. So possibly they may go for carbohydrate antigens too, but B1 B cell, yes, also the marginal zone B cells, yes. Response to protein antigen, yes, it is there for the protein antigens. Possibly B1 B cell can also have the protein antigen which can have this response, but marginal B zone B cells, yes, they can respond to the protein antigens. Memory cells, memory B cells, yes, follicular B cell or the conventional B cells had memory, whereas B1 B cell very little or none, which is unknown for the marginal zone B cells. The surface IgD on mature B cells, present on naive B cells, yes, which shows that these are the immunocompetent B cells which are ready to receive any antigen, whereas little or none in B1 B cell, little or none in marginal zone B cell. So these are the differences. So we are most of the time we are talking about this only, but yes, when you attend the JC or the seminars or come across of the paper, then you can get these terms also. So you should be aware of there being a PG student that yes, there are different kind of B cell pool and they have the major sites where they reside. So finally the last objective, B cell related clinical disorders. Although I think you have the immunodeficiency class separately, but I will just name two slides or three slides on this. The B cell immunodeficiency. It can be low production of one immunoglobulin or more antibody isotypes. It can be one or it can be more. Complete absence of mature B cells, plasma cells and immunoglobulin, to selective absence of only certain class of immunoglobulin can give you B cell immunodeficiency. Either you are having absence of mature B cells or the plasma cells or the immunoglobulin, or maybe one class of immunoglobulin. Patients with inherited B cell defects have recurrent bacterial infections. that display normal immunity to most viral and fungal infections because the cell mediated immune system is largely unaffected. Because T cells are there, so they are having cell mediated immune system, but yes, the inherited B cell defects have recurrent bacterial infection because opsonization will not be there. Okay, the B cell effector function. Okay, but cell mediated immune system will be there, so they can fight with viral and fungal infection, but bacterial infection will be there. Common infections are caused by encapsulated bacteria such as Staphylococci, Streptococci and Pneumococci. Because antibody is critical for the opsonization and clearance of these organisms. More common deficiencies are common variable immunodeficiency and selective IgA deficiency, which appear to involve multiple genes and then can have continuum or different kind of phenotypes. So this picture shows you that yes, the deficiency of B cell can cause 50% contributed by B cell deficiency. The cellular or T cell deficiency is 10%, complement system deficiency 2%, phagocytic deficiency 18%, which is by macrophages and other cells, and combined, that is B and T cell deficiency, both severe immunodeficiency, that is 20% out of this 100. So this picture, I think in immunodeficiency you will learn more, but I will just want to focus here the stem cell to lymphoid progenitor to precursor B cell to mature B cell. So here, if both B and T cells are deficient, we can have severe combined immunodeficiency, and if mature B cells are formed but common variable hypoglobulinemia is there, the gamma globulins are not formed, or selective immunoglobulin deficiency, that can be IgA deficiency or X-linked hyper IgM syndrome. These are the important clinical disorders related to B cell deficiency. So this is a list given in all the books. The severe combined immunodeficiency can be RAG1, RAG2 deficiency, ADA, PNP, JAK3 deficiency, IL-2 gamma deficiency, these receptor gamma deficiency, ZAP70 deficiency. These deficiency can cause different like no TCR or immunoglobulin gene rearrangement, toxic metabolite in T and B cells, defective signal from TCR if ZAP70 deficiency because it is involved in the signal transduction. So this is about severe combined immunodeficiency. When we talk about gamma globuliniemias, it can be X-linked A gamma globulinemia where no gamma globulins are formed, Bruton's tyrosine kinase is defective, no mature B cells are formed. X-linked hyper IgM syndrome where defective CD40 ligand, common variable immunodeficiency that can have low IgG, IgA or variable IgM, selective IgA deficiency where low or no IgA immunoglobulins are formed. So I will discuss only two, the X-linked A gamma globulinemia or the Bruton's hypogammaglobulinemia, which is characterized by extremely low IgG levels and by the absence of other immunoglobulin classes. So babies born with this disorder have virtually no peripheral B cells and suffer from recurrent bacterial infections. So X-linked A gamma globulinemia is caused by a defect in Bruton's tyrosine kinase BTK, which is required for the signal transduction through the BCR or B cell receptor. Without functional BTK, B cell development, besides without functional BTK, B cell development in the bone marrow is arrested at the pro-B to precursor B cell stage, and the B lymphocytes remain in the precursor B cell stage with heavy chains rearranged but no light chains in the germline configuration, because we have learned that from progenitor to precursor B cell there is a gene rearrangement, first of the heavy chain then of the light chain. So heavy chain gene rearrangement is there, but no light chain gene rearrangement in the germline configuration. So present day they have some antibiotics and replacement therapy in the form of passively administered antibodies, which can make this disease quite manageable. Then we have common variable immunodeficiency disorder, where patients or CVIDs do share recurrent infection resulting from immunodeficiency marked by reduction in the levels of one or more antibody isotypes and impaired B cell response to the antigen. This condition can manifest in childhood and later in life, when it is sometimes called the late onset gamma globulinemia. People have respiratory tract infection by common bacterial strains, and the most common symptoms and can be controlled by administration of immunoglobulins. Most cases of CVID have undefined genetic causes, and most patients have normal numbers of B cells, suggesting that B cell development is not the underlying defect in these cases. And last, the selective IgA deficiency. We know that immunoglobulin A, IgA, is present in the secretion. This is the secretory antibody. So most common, effective approximately 1 in 700. Individuals typically exhibit normal levels of other antibody isotypes, have full lifespan, but the IgA deficiency is there. If IgA deficiency is there, what will happen? It will be troubled only by infections of the respiratory and the genitourinary tracts, and which is the primary sites of IgA secretion. Family association studies have shown that IgA deficiency sometimes occurs in the same families as CVID, suggesting some overlap in the causation. The spectrum of clinical symptoms of IgA deficiency is broad. Most are asymptomatic, whereas others may suffer from an assortment of disease complications. And the defect in IgA deficiency is related to the inability of IgA expressing B cells to undergo normal differentiation to the plasma cell stage. And gene or genes outside of the immunoglobulin gene complex is suspected of being responsible for this family fairly common syndrome. So in this, the B cells undergo normal differentiation to the plasma cell stage, but again the IgA antibodies are not finally formed. So with this, I would like to thank you all, and just to summarize that what are the important points which we have covered today. Today's class, we have covered the B cell development, their maturation, their activation and their differentiation. Then how these B cells can be activated by thymus independent antigens and thymus dependent antigens. What are the different events takes place when thymus dependent antigen activates the B cell, and these B cells further start producing plasma cells, and these plasma cells can form immunoglobulin as well as they form the memory cells. Then once these cells are activated, immunoglobulins are formed. They have recognized these antigens, and these antigens which have been taken care by the immunoglobulins will be finally disposed of by effector functions. So humoral immune response effector functions are important: the complement activation, the opsonization, or the ADCC, depending upon the type of antigen that is there and what is a kind of pathogen is there. So then this pathogen will be killed and it will be cleared off from the system. Then once the antigen is activated, the antigen is activating B cell, immunoglobulins are formed, humoral immune response is there. Then we can have primary response as well as secondary response. If a person has come across with the antigen for the first time, it will have primary response. The same antigen when given second time, then we can have heightened response. We have learned the differences very clearly about these primary and secondary response at different events. Then we have covered after that we have covered the different B cells, that we have B2, V1 and marginal zone of B cells. These B cells, what are the differences? The conventional B cells are B2 B cells. And then finally we have learned about some of the clinical disorders which are linked with B cell immunodeficiency. With these, I would like to close this lecture, and again I request you all, if anything is not clear, you can always come to my room after this corona 19 or COVID-19 problem is over. We can discuss in detail anytime when you are coming. Just fix up the time when I am free. Thank you once again.